This UCSD TV program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest programs.
We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. CARTA brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
So as we all know, mammalian skin is decorated with diverse appendages, including mammary glands and hair follicles with their associated sebaceous glands, and apocrine glands, which are found in hair follicles mainly in the axilla regions in humans, and also eccrine sweat glands, which play an extremely important regulation in human thermoregulation. So the questions I'd like to address in this talk are, firstly, how did these organs evolve? And secondly, do they have a common evolutionary origin? For the most part, skin has not left us with a fossil record, and so this makes it very difficult to answer these types of evolutionary questions. So one approach to addressing these issues is to ask whether the development and genetics of these organs can give us some clues to their evolution. So during embryonic development, it turns out that the very early stages in the formation of mammary glands, hair follicles, and sweat glands are morphologically very similar. So the mammary gland develops from the ectoderm, which this is the outermost layer of the embryo, and it's diagrammed here in pink, and underlying mesenchymal cells, which are shown as black dots. And the first sign of glandular development is a localized thickening in the ectoderm, which is called a placode. And this then buds down into the mesenchyme and induces the formation of a fat pad. After that, the mammary sprout continues to grow downwards and starts to branch and develops a lumen. If we look at hair follicle development, we find that this is also characterized firstly by the formation of a localized thickening in the ectoderm, followed by formation of a bud. And after that stage, the development of hair follicles diverges from that of mammary glands. The last organ to develop in embryonic life out of these three is the eccrine sweat glands, which form around birth. And these also form first through thickening of the surface ectoderm, budding into the mesenchyme, and then further downgrowth, which in this case involves curling of the tube to form the gland and the development of a lumen. Another thing that these organs all have in common is that we know that it's extremely important that the cells of different types are able to communicate with each other. So in each case, the epithelial and mesenchymal cells engage in extensive crosstalk between these different cell types. So this crosstalk is made possible by the secretion of signaling factors from one cell that are then received by another cell and cause changes in this receiving cell that can include changing its proliferation status or its fate. Two of the really important cell-cell signaling pathways that are involved at early stages of appendage development are the Wnt signaling pathway and the ectodysplasin or EDA signaling pathway. In each case, these involve secreted Wnt ligands that are received by a receptor. In the Wnt pathway, the ligand is known as Wnt. So this is made by another cell, and it floats out and then starts to interact with receptors on the receiving cell. And this causes a whole series of intracellular changes that end up with the activation of target gene expression. And these genes then can cause a change in fate of the cell. Ectodysplasin signaling similarly involves a secreted ligand, in this case called EDA, and this interacts with its receptor and causes a different set of changes intracellularly, but these also result in expression of target genes. So it turns out that Wnt signaling is absolutely key for the formation of all of these different appendages. In its absence, they completely fail to form. And if you overactivate this pathway using genetic tricks in mice, we see that you get extra appendages forming. Downstream of Wnt signaling, the ectodysplasin pathway is activated. And this requires Wnt in order to get started, but then takes over from Wnt in further promoting the formation of these organs. So both the similar morphology and the similar molecular pathways involved in the formation of these diverse organs suggest that they may have a common origin. If we look during evolution, our ancient ancestors, the sauropods, at least some of them were thought to have a glandular skin. And this was selected for in the therapsids. And these animals lacked scales, in fact, and had completely glandular skin. And people have argued that one of the functions of this glandular skin was to provide moisture through skin secretions to the eggs of these creatures, which are known to have been very thin-walled and would likely have dehydrated otherwise. The development of hair has been argued to be possibly a specialization that could have aided in the delivery of glandular secretions to eggs. So the mammals that exist today fall into several classes, of which the monotremes still lay eggs, and they lack nipples and suckle their young via specialized hairs. And these animals include the spiny anteaters and platypus. Whereas marsupials do have nipples, but these nipple primordia actually co-form hair follicles and mammary glands during development, as I'll show you in a minute. The placental animals, which include ourselves, have developed hairless nipples, and these probably facilitate the suckling of newborns. So there's also some molecular evidence for this kind of a scheme. So it was thought that the mucus-secreting surface epithelia make antimicrobial enzymes as an important function of these epithelia, and these include xanthine oxidoreductase and lysozyme. And as glands developed in order to more effectively secrete these antimicrobials, and eventually forming mammary glands, at the same time as this development or evolution occurred, the gene encoding lysozyme actually became duplicated. And one of the copies evolved into the alpha-lactalbumin gene, and this encodes an important component of milk and is also involved in the manufacture of lactose, another important component of milk. It's also been shown that xanthine oxidoreductase has a secondary function in addition to its antimicrobial properties. It's also important in encasing fat droplets in milk and allowing their secretion. So these enzymes seem to have adopted additional functions that allow them to help in milk production. Another piece of evidence that for a common origin for mammary glands and hair follicles came, as I already mentioned, from studies of marsupial mammary development. So in marsupials, the nipple develops, as I mentioned already, from a thickening in the ectoderm, and this elongates and develops two types of sprouts, a primary sprout and secondary buds. And these secondary buds go on to form mammary glands, whereas the primary sprouts develop into hair follicles with associated sebaceous glands. So this is all happening during the embryonic development of the marsupial. And at the last stage of development, the nipple actually everts and it retains these sebaceous glands and, of course, the mammary glands, but the hair follicles degenerate in most marsupials. Although it is known that koala bears actually have hairs still emanating from their nipples. So this common development of these different types of organs in the same structure really provides support for the idea that these things may have co-evolved. So, of course, the numbers and locations of mammary glands have evolved to fit the litter size and feeding position. So in humans, we generally have one baby and we have two mammary glands. The babies we usually hold our babies to our chest, and that's where the mammary glands are located. By contrast, pigs have very large litters with an average size of 10, and they have 12 to 14 nipples or teats. They tend to feed in a lying position, and their nipples are located all along the side of their body. And cattle generally have a litter size of 1 and only 4 nipples or teats, and they feed standing up with the mammary teats in an inguinal position. So what are the mechanisms that may have permitted this adaptation of mammary gland numbers and locations to fit the needs of these species? So in many mammals, the embryonic mammary glands actually form along a line between the fore and hind limb buds in the embryo, and this is known as the milk line or mammary line, and it gradually becomes fragmented, resulting in the formation of mammary buds. And this is diagrammed for a mouse embryo. We can see something very similar in the rabbit embryo, and this is a scanning electron microscopy that allows us to actually visualize this raised mammary ridge. And this can also be seen in human embryos. So as I mentioned, the Wnt signaling pathway plays key roles in the initiation of mammary gland development. So there's no mammary development without this signal. And if we overactivate it, you can get extra mammary glands. And we can visualize this nicely by looking at one of the Wnt ligands called Wnt10b. And here we're using a technique called in situ hybridization to look at where the Wnt10b mRNA is being made. And this appears as a reddish-brown stain. So we can see this stains all along the milk line and is elevated in the developing mammary buds. And interestingly, in mice which lack a secreted inhibitor protein, which would be expected to result in excess Wnt signaling, we find that these develop extra nipples and extra mammary glands. So these findings suggest that changes in the pattern of Wnt signaling activity could underlie the adaptation of mammary gland location and number. So like the mammary gland, hair and sweat gland formation varies greatly between mammalian species, but also varies within species. So if we think about humans, of course, we have nice long hair on our head and very short, fine hairs over the rest of our body. But within our species, the hair can vary greatly in its texture and in its thickness. Our apocrine glands, as I mentioned, are mostly axial, and we have eccrine sweat glands all over our body. By contrast, a polar bear has hair that's of rather similar length over most of its body, but this hair is specially adapted and has large air pockets that allow the much better thermoregulation for the polar bear in cold conditions. It also, interestingly, has apocrine glands on the soles of its feet and leaves a scent trail with these. And its eccrine glands are confined to the soles of the feet, unlike in humans. And horses apparently completely lack eccrine glands, and instead they sweat by a different mechanism that utilizes apocrine glands, which are located all over the body of the horse. And when these are active, you get this lathering appearance that we see, for instance, in racehorses. And the hair of the body is mostly, the hair of the horse is mostly short, but has specialized appearances on their tail and the mane, where it's very long. So there's an incredible amount of variation here. For both hair follicles and eccrine sweat glands, the ectodisplacin signaling pathway plays very key roles at early stages of development. And indeed, loss of function of this pathway in humans leads to syndromes where there's really an absence of all these organs. More relevant to evolution, though, it turns out that there's a variant of the receptor for ectodisplacin, which is associated with increased activity of this receptor. And in Asian populations, this variant receptor is specifically seen in people who have thick, straight hair and increased numbers of eccrine sweat glands. So particularly in the Li and Han populations, this variant is extremely predominant. And this is illustrated here using it, looking at Asian populations, and individuals who have either straight or non-straight hair. And you can see that if you have the 370A variant of EDAR, you're much more likely to have straight hair, whereas individuals with non-straight hair have a more or less equal chance of having either the A or the V variant. We can model this in mice by genetically replacing the V variant of the gene with an A variant. And this, in mice as well as in humans, results in the formation of thicker hair and also in an increased number of sweat glands. So see, here we're looking at the foot pad of a mouse, of actually several mice, and these are stained with blue stain to reveal the glands. And you can see the greatly increased numbers of these glands in the A variant. So mutation of EDAR to a more active form may underlie a subset of human variation in hair thickness and straightness and sweat gland density. So just to summarize what I told you, evidence from development, genetics, and evolutionary studies suggests that mammary glands, sweat glands, and hair follicles may be derived from a common precursor. The function of this precursor may have been to hydrate and protect thin-walled eggs, and later developed nutritional functions as well. Mammary gland evolution preceded the birth of live young. And mammary gland adaptation may have occurred in part through altered control of Wnt signaling, whereas variation in hair thickness and sweat gland density are associated with altered ectodisplacin signaling. Now, there's an awful lot that we don't know. Some very straightforward questions, such as what controls the formation of different types of appendages in specific regions of the body? How and why did human eccrine glands become ubiquitous over our body and intermingled with hair follicles? And very straightforward questions like why is human scalp hair longer than body hair? And of interest to many men in the audience, why do many human males lose their hair in a particular pattern? And I'm sure you can think of many additional questions that we really don't know the answers to yet and would like to. So just to reiterate that in future studies, because of the relative dearth of fossil records for mammalian skin, these types of developmental and molecular approaches to skin evolution can be particularly useful. And this is just my lab, who contributed to some of the work that I showed you. And thank you for your attention.